So today we're going to learn about the submandibular gland. In the submandibular gland, the first thing that you should know is the serous acini, the mucous acini, and the ducts. In the end of this video, I will give you the summary or the point of identification. If you get the slide of submandibular gland in your exam, so you can write this point there. So before just knowing the points, just get a concept of the submandibular gland, that how it will look like under a microscope. So here is the first picture, you are seeing it. So how the ducts will look like under the slide. So you can see here, this is a picture of the duct, this is another duct, and they are lined by the cuboidal type of epithelium. So if you see such structure, then these will be the ducts. Now the darker staining round structures are here. So these are the serous acini. Remember always that which type of staining is used. The serous acini will be darker and the mucous acini will be lighter. The stains that are usually used are using amoxicillin. So these two stains are basically used, and in that the serous acini will be darkly stained and the mucous acini will be lightly stained, right? So now you can get the idea that how serous acini will look like. These are the serous acini, and the lighter ones are the mucous acini and the ducts. Why these three things are important, you will just get to know in the end. So we'll move ahead toward the next picture. So this is another picture, and in this picture there is no marking, so now you can identify the duct is where. So here is the duct. Okay, the darker one will be serous acini, right? And the lighter one will be the mucous acini. Another specificity of the submandibular gland is that there will be serous demaloons. Now, what are serous demaloons? So the serous demaloons are that you will have a mucous acini and it will have a cap of a serous acini. So you can see here, this one, you can see this is a mucous acini and it has a cap of a serous acini. So basically it will be a mucous acini and it will have a cap of a serous acini. You can see another example is, find out another example in this figure. So you can see this is a mucous acini and this is a cap, right? This is a mucous acini and this is the cap, and these are the serous acini, right? These are wholly solely mucus, these are wholly solely mucus. Okay, this is a serous acini, a mucous acini, and it is having a serous acini over it. So a mucous acini having the cap of the serous acini will be called serous demaloon. So now we know about the ducts, the serous acini, the mucous acini, right? The mucous acini and the serous demaloons here. Moving ahead to another picture. This is another picture. So here you can see the lighter ones are the mucous acini and the darker ones are the serous acini. These all darker ones are serous acini. These two are the ducts. Now you can identify this one is a duct, this one is a duct, right? So you can notice another thing is that we have a lot of serous acini and a bit of the mucous acini. So this is another property, that the submandibular gland will have more or numerous serous acini and less mucous acini. Moving ahead to another picture. Okay, a bit blurred, but you can get an idea that these darker ones are the serous acini, the lighter ones here are the mucous acini, and now you can see here, this one is a serous demaloon, this one is a serous demaloon, right? This one is a serous demaloon, this is a duct, this is a duct, right? And if you see a duct-like thing but it has a central reddish thing, it means that the red blood cells are there. So these will be vessels, so don't mess up the ducts with the vessels, right? So that was all about the submandibular gland, and now I am going to give you the summary. So the summary is here. So what will you write in your exam if you find out a question to write out the point of identification or to identify the slide that is showing under microscope. So you will write the points of identification is that the submandibular gland is having a serous and the mucous acini, but the serous acini will be numerous. The second point is that you will have a serous demulons there and intercalated ducts will be there. 3 points are enough. So thank you so much for watching. Subscribe for more.